look at our immune system. It protects us from infections. It comprises a great diversity of cells and molecules with different functions, and they can be found all over the body. A fundamental property of the system is its ability to discriminate between self and non-self, so that viruses and bacteria can be attacked. And indeed, innovative basic research in tumor immunology during the last 50 years have revealed a number of ways whereby cancer cells can also be recognized as non-self. This led to cure of cancer in many mice and also to successful treatment of some patients. However, it was impossible to develop efficient, generalizable therapies for clinical practice. Clearly, an important piece of the puzzle was missing, and this is what our laureates this year have solved. Now, let us take a closer look at the T cell, with the T appearing here, and we focus on it. In the 80s, scientists had discovered T cell receptors that bind to structures recognized as non-self. You see the structure as a dot in purple here. These structures can be picked up from cancer cells and be presented by so-called antigen-presenting cells. T cells that recognize something foreign with the receptor, as here, they get a signal to act, but this is not sufficient to get them going. Additional proteins on the surface of the T cells must also engage in the decision, acting as accelerator or gas pedal. They were called co-receptors, and the research field around them was termed co-stimulation. Many researchers contributed and observed that even more molecules were involved, but with quite the opposite function. They instead worked like brakes. And here they are in yellow, or at least one of them in yellow. And this one is called CTLA4, a complicated name, but please remember it, it will be very important.